Okay, so hello and hi everyone. Today I want to present to you that the structure of synapse. You need to know the structure of synapse before we move to the impulse formation across the synapse. So what you need to know is the synaptic knob, which is this part, and it is content of numerous mitochondria. It is to supply energy for the activities of neurons. Next, it is membrane bound synaptic vesicle, which is this part, this little thing. It is membrane bound synaptic vesicle. It is contain neurotransmitter, which is a chemical messenger involved in the transmission of impulse across the synapse. So yes, and then next you need to know the presynaptic neuron, which is a nerve cell that carries a nerve impulse toward the synapse, which is this neuron. This is what we call a presynaptic neuron. This neuron need to pass this into the postsynaptic membrane. That's why we want this name like presynaptic neuron. After it pass to this, it will become postsynaptic neuron. Yes, that's it.
Okay, so next I will explain. Next is synaptic cleft. So what it means of synaptic cleft? So synaptic cleft is actually a narrow gap that which separates postsynaptic cell from the presynaptic cell. So the size of synaptic cleft or the width is around 20 nanometer, and is quite small. So next, we enter postsynaptic neuron. What is exactly postsynaptic neuron? Postsynaptic neuron is a nerve cell that carries a nerve impulse away from the synapse. So postsynaptic neuron contain ligand-gated ion channel on the postsynaptic membrane. So this is the ligand-gated ion channel, as you guys can see. So the ligand-gated ion channel has a specific receptor for neurotransmitter. See, you can see the neurotransmitter can pass through the ligand-gated ion. So the other function of the ligand-gated ion is to allow movement of ions into the postsynaptic membrane, to allow ion movement right here. Lastly, the synaptic vesicle. So as we know, vesicle containing neuron transmitter, as you guys can see here. So there are two main neuron transmitters. So the first one, we call it as acetylcholine. Acetylcholine is actually secreted by parasympathetic neuron, and it is degraded by acetylcholinesterase. Next neuron transmitter is norepinephrine prime. It is secreted by sympathetic neuron, and it is degraded by monoamine oxidase. So that's all you guys need to know about the structure of synapse.
Alright, moving on into the impulse transmission across synapse. So when action potentials arrive at the axon terminal, as you can call synaptic knob of the presynaptic neuron, which is around this one, and this one would be the postsynaptic, the presynaptic membrane will undergo depolarization. So this depolarization will trigger the opening of voltage-gated Ca2 plus channels. So what's the function of this is that the presynaptic membrane will become more permeable to Ca2 plus, and Ca2 plus will be able to diffuse into the synaptic knob from extracellular fluid. So when there's influx of Ca2 plus in here, it will trigger the synaptic vesicle to fuse with the presynaptic membrane and release the neurotransmitter to the synaptic cleft via exocytosis. So exocytosis is that it will just dump out the content. So as you can see here, the neurotransmitter is now across the synaptic cleft. Then it will bind into the specific receptor. This is called the ligand-gated ion channels. So this binding will cause the channels, the ligand-gated ion channels, to open on the postsynaptic membrane. So when this open, this will allow, for example, Na plus to diffuse through, and this will trigger the depolarization on the postsynaptic membrane. So when depolarization occur, it will generate excitatory postsynaptic potential, which is the EPSP. So when the depolarization is above threshold level, the action potential produced is at postsynaptic membrane. So first, as we know just now, the action potential starts from here, right? So now that it's produced here, basically it's like transferred down here. So after the action potential at postsynaptic membrane is produced, the neurotransmitter is then removed. So there are two ways to remove, which is firstly, it's very simple. It's basically that the neurotransmitter are actively transported back into synaptic knob that releases them. So basically the neurotransmitter here, it goes back inside here via active transport lah. And then the second method is the enzymatic degradation by specific enzyme in the synaptic cleft. So for example, this is the acetylcholinesterase degrades the acetylcholine, and the monoamine oxidase degrades the norepinephrine.